Welcome back to Acing AP Biology. In this lesson, we will be going over the regulation of gene expression, particularly in prokaryotes. Both prokaryotes and eukaryotes have multiple ways of controlling gene expression. In eukaryotic cells, gene expression is regulated based on internal and external factors, along with cell specialization. Some genes may be available for expression in some cells, but not in others. And gene expression can be enhanced under certain circumstances. Alteration of DNA also affects gene expression. DNA in eukaryotes contain large areas of non-coding sequences called introns, which allow for variable gene expression. Prokaryotes primarily use operons for gene regulation. Operons do not exist in eukaryotes. An operon is described as a segment of DNA encompassing a set of genes, an operator, and a promoter. The operator is near the promoter and regulates access of RNA polymerase to the genes. The promoter is where RNA polymerase binds. The genes on the operon are also known as clustered genes. Now let's discuss the difference between repressors and inducers. These are both proteins that are involved with operons. Regulatory genes code for repressor proteins, which are allosteric, taking both active or inactive forms. Repressors in the active shape bind to the operator and block transcription of genes. Inducers attach to repressor proteins and inactivate them, so transcription can take place. So basically, repressors turn off gene transcription and inducers turn on gene transcription. Now let's discuss negative and positive gene regulation in prokaryotes. There are two forms of negative gene regulation, repressible and inducible operons. Repressible operons are normally on and transcription can occur. This is because the repressors are usually in their inactive form. However, when they are activated, usually by a co-repressor molecule, they bind to the operator and transcription stops. Inducible operons are normally off, and transcription cannot take place because the repressor is active and attached to the operator. An inducer can bind to the repressor, inactivating it and turning the operon on, allowing for transcription to take place. The lac operon and trip operon are two forms of negative gene regulation. Positive gene regulation includes operons that are involved with activators, which are allosteric proteins that promote transcription. E. coli bacteria utilize positive gene regulation for their metabolism. When glucose levels are low, transcription of operons that carry genes for other metabolic processes is amplified. Cyclic AMP builds up when glucose is low, and it binds to the activator CAP, making CAP more active. Active CAP now binds to the promoter region and helps the RNA polymerase attach to the promoter, thereby triggering transcription. The lac operon is a type of inducible operon. Therefore, it is normally off and is catabolic. The regulatory gene codes for an active repressor, which then binds to the operator. The lac operon contains genes to code for enzymes that can break down lactose. When glucose is low, lactose, another sugar, can be used for metabolism. However, when glucose is available, the breakdown of lactose is not necessary, and so the lac operon is usually off. When glucose levels decrease, an isomer of lactose, allolactose, binds to the repressor, acting as an inducer. This changes the shape of the repressor, making it inactive. Now the operator is free, and RNA polymerase can transcribe the genes on this operon. When glucose levels increase, the inducer releases, and the repressor becomes active again and binds to the operator, thereby blocking RNA polymerase. The lac operon is also regulated positively with the CAP activator, as I mentioned earlier. The trip operon is repressible and therefore is on most of the time. It is normally anabolic. The regulatory genes produce an inactive repressor. TRP stands for tryptophan, which is produced by E. coli and regulated by the TRP operon. The genes on this operon code for enzymes that catalyze tryptophan production. When TRP levels get too high in the environment, the TRP operon is turned off. TRP molecules act as a co-repressor and bind to the inactive repressor, activating it. The repressor can then bind to the operator and transcription is shut off. Once tryptophan levels decrease to a more balanced state, the co-repressor releases and the operon is turned on once again. That's it for operons. In the next lesson, we will be going over ways eukaryotes regulate gene expression. See you then. For more bio content, hit subscribe.